It's time for 5 and 5. Here's a 5-minute breakdown of AP Bio Unit 1 concepts you absolutely need to understand to help you get a 5 on your AP test. 5 minutes starts now. The real MVP. First up, water. More than a drink, it's the MVP of life. Water's polar, which means the electrons that make it up aren't shared evenly. That polarity lets water form hydrogen bonds, and that gives water its superpowers. High specific heat keeps the body temperature stable. High heat of vaporization makes sweating and evaporative cooling possible. Cohesion, adhesion, and surface tension all allow water to move from the roots of trees to the leaves at the top. Without these properties of water, biology as we know it just wouldn't work. So besides water, what else is important? The next segment, build or break. Biological molecules, the real building blocks, they're mostly built of carbon, nitrogen, oxygen, and hydrogen atoms. Sometimes sulfur and phosphorus join the party too. Biological molecules are like Legos. The individual building blocks are called monomers, and the constructed set is a polymer. Two reactions are involved in breaking and building these blocks: hydrolysis and dehydration synthesis. Hydrolysis adds water molecules to break polymers into monomers. Think digestion. Dehydration removes water to build polymers. Repeating dehydration synthesis over and over again gives you polymerization, the formation of large macromolecules. These macromolecules fall into 4 categories. This leads us into our next segments. Carbs. Large carbohydrates or polysaccharides are built by joining together monomers called monosaccharides. Polysaccharides store energy and give structural support in the cell, sort of like a battery. But carbs aren't the only ones running the show. The next macromolecule, nucleic acids, DNA and RNA. These run biological systems. DNA and RNA are made from monomers called nucleotides. A nucleotide is made up of a 5-carbon sugar, a phosphate group, and one of the 4 nitrogenous bases. But DNA and RNA have some key differences. DNA contains the 5-carbon sugar deoxyribose, while RNA contains ribose. DNA uses nitrogenous bases of adenine, thymine, and cytosine and guanine, while RNA has thymine replaced with uracil. DNA is typically double-stranded, while RNA is usually single-stranded. In DNA, because adenine pairs with thymine and cytosine pairs with guanine, the 2 strands are complementary to each other. The next macromolecule, proteins. Proteins are chains of amino acids connected by peptide bonds between the carboxyl group of one amino acid and the amine group of another. The interactions between the amino acids R group determines the protein's structure and therefore its function. Now onto the saturated lies, lipids or fats. Like other macromolecules, dehydration synthesis plays a role in forming these molecules. But they don't follow the classic monomer to polymer relationship. Lipids are mainly nonpolar, which makes them hydrophobic. Their functions can vary, but commonly include hormone formation, cell membrane composition and stability, and long-term energy storage. Lipids that contain fatty acid chains can either be saturated or unsaturated. Saturated fats have no double bonds and are generally solid at room temperature, whereas unsaturated fats have double bonds and tend to be more fluid at room temperatures. And yes, a lot of what you've heard about fats, saturated lies. And that's the rundown. Water, macromolecules, sugars, lipids, proteins, DNA, they all work together to build and sustain living systems. Bottom line, everything in your body is constantly being built up and broken down using only water. In under 5 minutes, we just went through the overview of Unit 1. Dive more into the specifics and get a comprehensive AP Bio review course. Visit collegeprep.uworld.com/AP.